Alrighty, good morning. We're going to talk about Unit 2, which is cell structure and function. There's 106 slides, so I'll try to go quick. There's a lot of pictures though. So most of this is easy by now because we've talked about it all year. So I'll go kind of glaze over some of the stuff. So hopefully that's okay. But the cell is the smallest unit to show properties of life. What that means is if we go smaller than a cell, it doesn't represent an entire organism. So if we have one cell, we can make a whole organism out of it. Think about like cloning and stuff like that. But if we only had a mitochondria, or if we only had a ribosome, that doesn't represent the entire organisms. That's talking about. There is stuff smaller, but it doesn't show the properties of life or an organism like we would think about. And so at the end of class, or at the end of these, we're going to talk about the membrane in more depth. But there's three big sections of every cell. A membrane on the outside, something that holds genetic, or there's an area of that holds the genetic material, and then sort of the gushy stuff or the liquidy stuff that holds it. So plasma membranes and cytoplasm are in everything. In a eukaryote, it's called a nucleus, and in prokaryotes are called nucleoid regions. So the organelles, if you translate that in Latin, it means little organs, and so it's just the little pieces and parts that is in it, and we'll talk about those as we go through. Two kinds of cells. Eukaryotes have a nucleus. They have membrane-bound organelles. We are eukaryotes. Prokaryotic cells are, they don't have a nucleus. They are little. They have a circular DNA. They are bacteria. So bacteria and prokaryotes are the same thing, and eukaryotes are everything else. So animals, plants, protists, and fungi. So just a list of the different organelles and what kind of cells they're in. Another picture. And so this is just talking a little bit more about having a nucleus, and EU means true, and carry means kernel. So it's like a true nucleus is the weird Latin how that translates. And then it just talks about what I talked about before with the four things that are eukaryotes. And then we have organelles because we have fancy things that we have to carry out, and so we have to change glucose into ATP. We have to get rid of wastes. We have to do different jobs within our cells. Bacteria aren't that fancy, and so they haven't evolved to need all that because they are simple. Again, just a list of stuff that's in eukaryotes and a little bit more. And then a picture of a plant cell. One thing that plants have that animals do not is a large central vacuole, primarily to hold water. And then that's how they maintain the shape of a plant and turgor pressure, and sort of when a plant is wilty or not wilty, it's because that central vacuole might be empty or full of water. Just another example of a eukaryotic cell. And then this is a white blood cell. Different cells they contain different things because they have different functions. And so that's just another example. And then there are 5 kinds of white blood cells. I worked in a lab before I was a teacher, and so I thought this was interesting to know. And so there's 5 kinds, and they do different jobs, specific jobs. So one is like if you have allergies, you have more of a certain kind of white blood cell. If you have, if you get a bacterial infection, you use a different your body uses a different kind of white blood cell. So just a little added thing. And so in the nucleus, it contains our DNA. It doesn't go out into the cytoplasm ever. We'll talk about transcription and translation later, but it stays in there, and then it is contained within a membrane called the nuclear envelope. Sometimes you'll see it called a nuclear membrane. It's all the same. And so again, with this has a few more things in it that we'll talk about, but just a nice list. It's a picture from your book. Another picture, another picture. And then the nuclear envelope is just the membrane that is around the nucleus. So just exactly what you might think. The nucleus has pores in it. They're called nuclear pores. That's how the messenger RNA gets in or gets out, and then other things get in to do the different jobs or whatever they might need to get in and out of. But it's just a way to control what goes in and out. And so one of the interesting things that it talks about in this slide is it says the outer bilayer is continuous with the endoplasmic reticulum, which we'll talk about later when we talk about the endomembrane system. And so this is just some pictures of what they look like with a really cool microscope, and then just a little drawing. And then just some vocabulary. The nucleoplasm is the cytoplasm of the nucleus, and then the nucleolus is a special little area of the nucleus where they make parts of ribosomes. It's sort of the chicken and the egg of the nucleus makes protein, or the ribosomes make proteins, but where did the initial stuff to make the ribosomes come from? And so that all comes from the nucleolus. Where does that come from? And way back, it comes from the cytoplasmic parts of your mom's egg. And then this is the same stuff, different forms. So chromatin is the loosey, disorganized stuff of genetic material, and then chromosomes is when we coil it up and then we make an X-shaped something. So when in class, remember I said if you take a rubber band and you twist it and twist and twist and twist, it'll sort of bunch and coil up on itself. That would be a chromosome, and then the disorganized sort of loose rubber band would be like the chromatin, or I think of it like spaghetti, and then like spaghetti wound around your fork or stuff like that, just sort of to visualize it. And then that is a condensed one. They usually look like an X. So when we drew them in class, we would draw them like X's. Sometimes we draw them like sticks. If that's what they look like, that's why we draw them like that. And then other things that we need, eukaryotes, we have plants and animals, protists and fungi. We mostly talk about animals and plants. We have mitochondria because that's how we convert our energy. Plants have both because they need to, because they have that, and plastids like chloroplasts, and then they can convert sun's energy, and then they also have mitochondria because they have to do cellular respiration as well. So mitochondria, we'll talk about sort of the how we use it when we get to cellular respiration, but structurally and sort of other things that go along with it's on this slide. It has inner and outer membranes. So if you've ever seen it, it sort of looks like a kidney bean, and then it has like squiggly little inner membranes. That inner membrane is so that we can build up a gradient during cellular respiration. And so that'll, if it doesn't make sense, it'll make more sense later. And then one really cool thing is the mitochondria has its own DNA. So way back at the beginning of time, the theory is that mitochondria was sort of like a bacteria-like something that could convert glucose into usable ATP, and then we sort of grabbed onto it. Early eukaryotes grabbed onto it and said, "Ooh, that's useful," and then we have incorporated it into us. So now mitochondria relies on us, and we rely on mitochondria, and that's called endosymbiosis. We took it into our cells, and we have a symbiotic relationship. So just some pictures of the inner and the outer membrane. Plastids are in plants, some fungi, some protists, and they are things that have color. We care the most about chloroplasts, and then same sort of thought is there are some bacteria that are photosynthetic, and so the sort of thought or the theory is that early plants incorporated that into their cell, and now they live inside the plant cells, and then the plants rely on them, and they rely on the plants, and so endosymbiosis in a plant. And then just some pictures of some cool chloroplasts. The pigment, the green, is chlorophyll, and the thing that contains it is a chloroplast. I remember it because like plastic is like a plastic container, like Tupperware or like Rubbermaid or Gladware. So the plastic that holds it is the chloroplast, and then the chlorophyll is what fills it up. And then just another picture. And another picture. Top is bacteria, bottom is eukaryotes. And so this one would be a plant because it has a cell wall, it has chloroplast, it has a large central vacuole. Just to look at it and try to identify. And then prokaryotes are small. There are more different kinds, species of prokaryotes than there are all other eukaryotes combined. So there's more bacteria than there are plants, animals, protists, and fungi all combined, which is sort of mind-boggling, but it is. And then they have different shapes and sizes, and some are round, some are rod shaped, some are spirally. And so it just depends. Alrighty, so then some stuff about prokaryotes. Some of them have cell walls, some of them don't. And so if you were to stain them, some of them would be purple and some of them would be pink. And so we could look at them under microscopes and figure out it's called gram-positive or gram-negative. It's a lab test that you could do, and then that helps us identify what they are, and then would help doctors to know what antibiotics to use and different stuff like that. It's all based on the presence or the absence of a cell wall. And then a flagellum is just like a tail. I'll show you a picture in a sec. And then pili helps cells move across surfaces. So it's sort of like helps them like scooch stuff. There's a thing called a sex pilus. It's sort of like a like tail-like thing that goes from one bacteria and then another bacteria, and then it transfers genetic material across. And then that's how we have genetic recombination between two slightly different bacteria that then can combine and then turn into something new and different and sometimes terrible. Alrighty, and so they have ribosomes because every living thing needs proteins and enzymes and stuff like that. They don't have a nucleus, but they do have a nucleoid region. The reason that it has a nucleoid region is because when bacteria go through binary fission, it has to ensure that the two, when it splits in half, that each one will get its own genetic material. So what happens is the circular DNA replicates, and then a little bit goes to each side of the cell, and so then it splits down the middle, and then it each has its own genetic material. And then plasmids are little chunks. We talked about plasmids when we talked about genetic recombination and when we talked about the biotech later in the year, but it's little chunks that you can move from bacteria to bacteria, and then or bacteria can move it from bacteria to bacteria, and then can change its genetics. And so the one example of that is antibiotic resistance, and don't even get me into hand sanitizer. So just a picture gives you a little, so the pilus is on there, the flagella is on there, all those little things that we talked about. Just some more pictures. And then archaebacteria is its own domain. There are three domains: archaebacteria, bacteria, and eukarya. They have characteristics similar to eukaryotes and prokaryotes, but enough sort of a mix that they became their own domain. We don't talk too much about them. And then we did a lab on this, which I'll explain in a different video. But cells are small because that's what works, and it's based on diffusion and then how much energy you need. And so we talked quite a bit about surface to volume ratio. And so the higher the ratio, so in this picture, a 3:1 to a 2:1, and then I think that's a 6:1. I don't know what it is. I can't do the math and I can't see it. The higher it is, so for this example, the 3:1 would be better than the 2:1, and that cell is smaller. Oh no, that would be that's 6, because look how big that cell is. That 6:1 doesn't even make any sense. Anyway, so smaller is better. because of diffusion. And we did the Auger Cube lab, and again, I'll explain that in a different video. And so for one of the important parts, and it seems like such a small part, but it's actually a super important part of a cell, is the cell membrane. And so we have what's called a phospholipid bilayer, and there are phospholipids. And in the last video, we talked about how it had a polar head and non-polar tail. And so then we would have, and then they'd fling in, and then we'd have a bilayer that goes like that. And then there's a whole bunch of stuff that's in it, proteins and cholesterol and other things that control what goes in and what goes out. Alrighty, so this is just a picture, and it shows sort of the inside and the outside of the cell with the lipid bilayer. It's not really a great picture, but there's a lot of stuff on the outside of our cells for identification, sort of like cells in our body have things on it that say to our body, you belong here. And so an autoimmune disorder would be like our body doesn't recognize that it's supposed to be here, so it attacks it. Or there's things on the outside of a bacteria that our bodies can identify as not supposed to be there, and then we can attack it. Or that cell or that organ is it has a receptor for a hormone or whatever. So there's a lot of stuff on the outside of a cell that's useful, usually for identification and reception, but there's a few other things too. And so the endomembrane system, I talked about this earlier. There is a continuous system of membranes from the nuclear membrane through the ER, through the Golgi, makes a vesicle, pops out through the plasma membrane. And so it's all connected because it's more efficient than sort of releasing stuff out into the cytoplasm and then having to go and grab it and contain it within membranes so that we could release it. So it's just how it works. And then we have within that, the first sort of stop after the nuclear membrane is the endoplasmic reticulum. There's two kinds. Rough has proteins or has ribosomes, so it can make proteins, and smooth does not have ribosomes, so it takes those proteins that were produced previously in the rough and then it makes them better. Sometimes it breaks stuff down, and then after that it moves on to the Golgi, and then it makes little packages that can be called vesicles or vacuoles. And so peroxisomes are ones that break down hydrogen peroxide and other things. Our body makes a lot of toxic things as a byproduct, and so we have to be able to get rid of that. And peroxisomes is one of the things that helps take care of that. And then lysosomes is sort of the digestive vesicle. Vacuoles are sort of, we need to contain some stuff. So maybe we broke some stuff down, but we don't want to just let that loose out into a cell. We sort of contain it until we can go over to the membrane and export it. And so it would get maybe picked up by the bloodstream, but it sort of holds it kind of like a little container. Central vacuole, like I said before, is in plants, and it primarily holds water, but it's huge. And so whenever you look at a plant and there's a huge, looks like an empty space, it's probably a plant. Golgi bodies is what comes after the endoplasmic reticulum, and then it finishes the job. It sort of it checks stuff and makes sure their purpose and packages them up and gets them ready for transport. Sort of like the proofreader, I guess, of the endomembrane system. And then we call our membrane selectively permeable because some stuff can get in, some stuff cannot. And then this is, we have hydrophilic, which means they love water, heads, and hydrophobic tails. So it would look like this in a really terrible drawing. But the hydrophilic heads and hydrophobic tails. And then when you put hydrophilic, hydrophobic things into water, which we are water-based, they do that. It's like an attraction and a repulsion. And so then we get a bilayer, which is pretty cool. The fluid mosaic model is how we describe it because it is a mosaic, like the picture of it changes. I always describe if you picture like a swimming pool with a whole bunch of ping pong balls on the top, and then they can sort of float around like that. And then if you throw in some pool noodles or throw in, I don't know, a beach ball, that would all sort of scooch around and change. And so the picture is a mosaic, so it's ever changing, but it's a fluid. And so it sort of represents what could be and is in our membrane. And so just a little picture of the heads and the tails and how they fling inward or outward. And then some stuff can get in and some stuff can't. And so there's a lot of controls. Some molecules can go right across if it's small, if it's hydrophobic, but some stuff can't. And so that we have to have proteins and different pathways of getting things into and out of the cell. And so this just shows us that little stuff can get across if it's small. If it's big, then it has to have help. And so that's where membrane proteins come in. And then this is just talking about transport proteins. It's like a little tunnel. So if you picture like a pool noodle and with that has a little hole in the middle, it might be kind of, if you picture it like that, it could pass through it. And then something can pass really easy. And then there's three ways that stuff get in. Passive transport, no energy, goes from an area of high concentration to low concentration. Active transport means it's going from an area of low concentration to high concentration or against the concentration gradient. And then endo and exocytosis, which both use the membrane to get things into or out of the cell. And so passive transport, there's two things. One is just diffusion. It just goes across the membrane from an area of high concentration to low concentration. Facilitated diffusion is when we have like a protein that it passes through. And so it's still going from high to low, but it has a helper. So the protein is the facilitator. And so it's called facilitated diffusion. And then just some pictures of like a little membrane protein. Looks sort of like a little tunnel or a little tube. And then active transport, it's going against the concentration gradient. So if you're pushing something against the gradient, so we're going from an area of low concentration to an area of high concentration, that's going to take some energy. It's going to take some work. And so it requires ATP. One of the examples of that is calcium pumps that have to do with our muscle cells and sort of our the nervous system. So it's just one of the examples. And then this is showing a little picture. So we're going from an area of low concentration on the bottom of the picture to an area of high concentration at the top of the picture, and so that would take some work. And then just another picture showing you the work. And then there are two kinds of membrane proteins that you should be familiar with. It's pretty easy, the vocabulary. Peripheral proteins are the ones that are on the, so if you think of your membrane kind of like this, they'd be on like the top, so the periphery. And then integral are the ones that are integrated, so they would go across the membrane, just exactly what the words sound like. And then there are 5 types of membrane proteins, and the name is what they do, so it's really pretty easy to remember. So adhesion proteins are the ones that help them stick to each other. So our skin cells aren't like bonded together, but they do adhere to each other. And so there's proteins on the outside. I sort of think of them like Velcro. And so they get next to each other and then they help the things stick together. Recognition proteins are ones that say, I'm a red blood cell, or I'm a white blood cell, or I belong here, or I'm a bacteria cell, I don't belong here. Receptor proteins are when we talk about cellular communication, so it's sort of like the ligand on the outside of the cell comes in, and then we recognize it, and then it goes and triggers the cellular communication cascade that comes after that, but we'll talk about that later. Enzymes, because sometimes you need to have a reaction in order to get something into or out of the cell, or recognize or start some sort of a process. And then transport proteins are the ones that are little tunnels, kind of like the pool noodle. So this is just a picture, and it gives you a just some examples. It's a nice picture. It's from your book. And then things can go across easy and some things can't if it's small. if it is water, if it is other things, it goes across. We'll talk about hypotonic and hypertonic in a second, but water goes across because water can go across. Sometimes salts or other molecules cannot, so the water balances out the concentration of the solute, or the sort of thing that's dissolved in the cell. And so diffusion is just from an area of high concentration to an area of low concentration. It's called going with the concentration gradient. It does not require energy. And so this is just a picture of it diffusing from an area of high concentration to low concentration. And then the rates of diffusion depend on five big things. How big it is, the temperature of the water, or the environment, I guess I should say, how steep the concentration gradient, how big the difference is between one side of the membrane and the other side of the membrane, or one portion area of the environment to another. If it's real close, it's slower. If it's real big, it's faster. The charge of the molecules and the charge of the environment, and then the pressure within the system. So if you're increasing pressure or decreasing pressure, that would sort of increase or decrease the rate of diffusion. So concentration is how many pieces and parts are in the fluid. So how much salt is in the water, how much sugar is in the water. And so then the concentration gradient is the difference between the two sides of the membrane. Tonicity, I drew a little picture. I did this on the board in class. But for this, it's not my best artistic work, but it never is. So this just shows, let me see if I can figure out how to do this with my camera. So this is just showing hypertonic. So we have a lot on the outside. And then if you compare the numbers, the water would go from an area of high water concentration to an area of low water concentration. So for the first one, would go from 70 to 10. So that would go from inside the cell to outside the cell. And so hypertonic is, it would shrink. And then this one is, we have a lot inside. So this is hypotonic. And so we would have a lot of water outside compared to a lot of water, or a little water inside. So then the water would go into the cell. So the cell would swell up and sometimes burst. And this, they're equal, so it goes into and out of the cell at equal rates. So it doesn't stop. It always continues to flow. So hypo, less on the outside; hyper, more on the outside; iso, same on the inside and the outside. And then osmosis is the movement of water from an area of high concentration to low concentration. Our cells, sometimes the salt or the sugar or the whatever particles can't get across, and so we have to move the water, and so then that swells or shrinks cells, which is sort of what I just showed you. Hopefully it made sense. And then just a picture of osmosis in semi-permeable membrane. We did a lab on this with sugar solutions. I'll explain that in a different video. And then turgor pressure is in cells that have a cell wall. When the water goes in and sort of swells up, in an animal cell it can burst. In a plant cell, it pushes on the cell wall, and the pressure that it pushes out on the cell wall with is called turgor pressure. And then osmotic pressure we did a lab about that, but I'll talk about that in a different video. But it's sort of the same thing. How much can it push or not push, I guess, when it comes to the pressure. And so just some pictures. The first one is regular old red blood cells, then some shrunk ones, then some swelled up ones, and then the last two are the same kinds of cells, but the inside is shrunk. And so just a comparison. And then whenever we bring things into and out of the cell, there's different processes, and then it's not small, it's not hydrophobic, it's not charged, it's not water, and so then we have to make little bubbles of stuff that can carry into the cell. And so we call this, there's two different things. Exocytosis is when we make a little bubble or a vesicle, and it goes over to the cell membrane, and then we make a little pore, and then the stuff scooches out, and the other one is we have some stuff and then we make a little membrane around it and then we bring it into the cell, so endo. And so here's just some pictures of those two things. Stuff going into or out of the cell. It's a cool picture. Hopefully it explains it better than my hand gestures. And then a real picture of it. And then just another picture. And so then there are 3 ways to bring things into the cell. The 2 easy ones are phagocytosis and pinocytosis. Phagocytosis is solids, so it's like eating, and pinocytosis is liquids, and so it's like drinking for a cell. Receptor-mediated endocytosis is when that thing that is sort of circulating around your body is supposed to come into a certain cell, and so it has to have a receptor on the outside that then it says, okay, you're allowed to come in. And so that's why it's called receptor-mediated endocytosis. That sort of has to pay the gatekeeper to get across. It's a little bit more specific because sometimes that's necessary. And just a picture of a white blood cell engulfing some bacteria, which is pretty cool. Then phagocytosis is that was solid, sorry. And then membrane cycling is when we bring it in or let it out. We sort of add some new or take out some old. So it cycles the plasma membrane, so it keeps it fresh because it's constantly happening. And then the cytoskeleton is just sort of structural stuff that helps the cells sometimes do jobs, sometimes be supported. And then there's 3 kinds that we talked about: microtubules, microfilaments, and intermediate filaments. And I'll show you pictures. So tubule is like a tube, and then it has certain subunits. So just it looks, to me, they look like exactly what they're called. And then a filament is thinner, no central sort of tunnel thing. And then an intermediate is sort of has different aspects, but it's not really either. So it's just a slightly different thing. So we call it something different. And then just a picture of a nerve cell. And then motor proteins are ones that help us move things. And so it would be active transport. And so sometimes we have to get the vesicle from the Golgi to the cell membrane. And so there's a little filament that runs across the cell and then it sort of walks along it. It motors across it to get it from point A to point B. And that's a little picture. There's GIFs on this on the internet if you want to look it up. And then flagella and cilia are hair-like things that are on some cells. And then we talk about it a 9-to-2 array. The reason that we know that they are associated with each other because they are, if you sort of chop them in like a cross, it's called a cross section, then it looks the same. And so it's a 9-to-2 array. And so that's just what it looks like. And then pseudopods are when a little part of the cell membrane sort of scooches out and then moves. Amoebas move using pseudopods. There's a picture, or there's a little video link there if you want to watch it. And then we have a cell, and then we have things that sometimes connect it, and then sometimes we're just like around, and then it makes different things. And so a cool thing that our body makes, it's called an extracellular matrix, which means it is some sort of meshy kind of structural thing outside of the cell. And so an example is bone is an extracellular matrix, and there are things within the bone that are living, so like bone marrow and cells and different stuff like that. They connect things, they hold things, they ligaments and all that kind of stuff, but the actual bone was a gushy kind of thing that then hardened and then became a really cool functional thing for our body. And then there are a couple kinds of cell walls. Just be familiar that there's different kinds of ones. Animals, we don't have animals when I say we don't have cell walls. Plants, fungi, some protists have different kinds, and then structurally, if it needs to be more rigid, then it has maybe a second one, because that's just what that thing needs. So think like bark on a tree versus like the outside of a mushroom kind of thing. So just some pictures. And then a cuticle is a like a extra layer. And so this talks about, we think of cuticles like on our like on our fingernails, but that's still a thicker protective thing. But it's on like the outside of plants, and it's on certain, like this says crabs and spiders, crunchy stuff, that it gets released, and then it's an extracellular matrix, it gets released, and then it hardens, and then it becomes a protective thing, kind of like a bone, but on the outside. And then just the cuticle on a plant. And then our cells hook together, and so that they can pass things between cells. This happens a lot with plants. It happens with us too. And then there are three kinds in animals, and then there's one big one in plants called plasmodesmata, or for animals, there's tight junctions, adhering junctions, and gap junctions. And so this just talks about the three of those. You can read through those, but it has to do with how we sort of have our cells next to each other, or we trade some stuff, or it's like a little passageway for molecules and different stuff we might need. Just a picture, just another picture. This has all of them in it, just sort of visually to help you out. And then the endosymbiont, or I call it the endosymbiotic theory, is, I talked about this earlier when we talked about mitochondria and chloroplasts. Way back, we weren't cells like we think of today, but there were all these little teeny tiny things. And so one of the cells grabbed, or a cell-like thing, precursor, grabbed a mitochondria, one grabbed a chloroplast, or something that's not a chloroplast or mitochondria like we think of them today, but they're precursors, and so then we sort of grabbed it and took it into, and then we rely on them and they rely on us. And so we think that because mitochondria and chloroplasts both have their own genetic material. And so there's, I'm sure, different reasons, but the one that makes the most sense is they have their own genetic material because they were their own genetically standalone thing way, way, way back, which is a pretty cool thing that we can sort of hijack them and take them, but they live within us, so they're happy to. So it's symbiotic. And then just a picture of the bacteria I was talking about. And then this is just a little bit of the evidence, which I talked about, but same idea. And then just a picture. And then last one, just another picture that they microscopically and genetically resemble things that used to be. So pretty cool. That's it.